Because of your security. Thank you. Oh no.
Wait,
Yeah.
does that work?
Let me.
Does that work now?
Yeah. Thank you.
Okay.
Because of your security. Thank you.
Yeah.
Oh no.
Okay.
Can I start to let people in?
Yeah, I think
All right,
so.
great.
Work now?
Yeah.
Okay.
Yeah. Because of your security. I think there's an echo there. Okay, so welcome everyone to today's Open Book Science Seminar. I'm very happy to have these two speakers today, Rio Sugimara from the University of Hong Kong and Yimin Chao, a PhD from the University of Hong Kong. Now she's a postdoc. And today we're gonna have this incredible seminar about immune engineering. And this seminar will be moderated by Leo Curian. Leo Curian is he did his PhD in genetics, and now he's working actively to try to understand the process of development, including RNA biology and development. So I'm very happy to have all of you here, and thank you for joining today and this great discussion. And I'm happy to give the floor to Leo for presenting today's speakers.
Thank you. I think I will start with Iming, as it is advertised. Sorry. Iming is PhD candidate from the University of Hong Kong from the lab of Rio. Am I getting it right, or you already finished your PhD and moved on?
I'm still doing PhD.
Okay, just making sure. She had a basic education in various parts of the world, as you can see, from including Shanghai, Hong Kong, and now she's a joint effort between the University of Cambridge and University of Hong Kong in the lab of Rio Sugimara. She's an accomplished PhD student. So far, she has managed to publish a couple of papers, a few preprints, and today she's going to talk to us about her latest work in Rio's lab.
Thank you. Maybe I can start now. Can you see my slides? Okay, thank you. Yes, thank you, Leo, for the introduction, and thank you the Open Box Science Seminar for inviting me. And I'm Yimin Chao, a second-year PhD candidate from the University of Hong Kong. And today I will share our recent accepted work and some ongoing project on the organoid-based single-cell spatial temporal gene expression landscape of human embryonic development and hematopoiesis. The hematopoiesis is the process of forming the blood cells in the body. It starts from a small population of the stem cell called hematopoietic stem cell. The HSC and multipotent progenitor build up the stem cell pool and can give rise to all the blood cell types, including the lymphocyte, myeloid cells, erythrocyte, and the platelet. Along this trajectory, the erythropoiesis is the process of producing the red blood cells. These cells are important for the oxygen transportation in the body. The erythropoiesis involves several stages, including the proliferation and differentiation from stem cells to the erythroblast, the production of hemoglobin, and maturation and release of erythroblast into the blood circulation. This normally happens in the bone marrow. But before the bone marrow take over this function, the yolk sac is an important site where the first hematopoiesis arise and support the embryo. In mouse, it is well known that there are 3 waves of hematopoiesis. The first and second wave arise in the yolk sac, which is called the extra-embryonic wave. It generates the primitive erythrocyte, megakaryocyte, macrophage, and lymphoid cell and support the embryo. Later, the intra-embryonic wave arises in the AGM region in the embryo proper and seeded into the fetal liver and give rise to all the blood sublineages. So nowadays, the studies are more focused on the intra-embryonic hematopoiesis wave, like how the endothelial to hematopoietic transition happen and how the specification of the blood lineages. And how about the extra-embryonic one? So in this study, this leads to our first question. Can we model the human early hematopoiesis in the extra-embryonic tissue? And to what extent do these extra-embryonic tissue regulate the hematopoiesis in human? We are adapting the expanded potential stem cells called EPSC that could give rise to both embryonic and extra-embryonic lineages in vitro. Compared to the normally used embryonic stem cells or induced pluripotent stem cells, these expanded potential stem cells have greater potential for the development. Theoretically, it can give rise to placenta, yolk sac, and all the cell types in the body. With this cell platform, we first established the human embryonic organoid culturing system from the APSC. We start from the pre-differentiation to get rid of the totipotency, and followed by the EB formation in the hanging drop. Later, we culture them in the stem diff commercial medium for germ layer patterning and hematoendothelial specification. This is how the organoid looks like, and it's like a balloon and mimic the yolk sac. To fully characterize the gene expression within the organoid, we conduct the time series single-cell RNA sequencing at these 3 time points. We also conduct the 10x Visium spatial transcriptomics on day 15 to study their spatial architecture. With this data, we found that hemo can generate multiple cell types from the extra-embryonic population like the yolk sac endoderm and trophoblast. Later, the other germ layers also come out, like the mesoderm and ectoderm. And finally, the hematopoietic population enriched, including the erythropoietic, megakaryopoietic, and the myeloid lineages. So this suggests that hemo can generate 3 germ layers and follow the developmental course as the embryo. For the spatial transcriptomics data, a huge limitation is the resolution of 10x Visium. As you can see here, in the margin of these HE staining slides, a single spot is occupied by multiple cells, nearly 10 to 20. So this limits our further exploration on the spatial architecture of organoid. So to solve this, we collaborate with a mathematician group. They designed a new statistic model to integrate the single-cell RNA sequencing data and the spatial transcriptomics data to obtain the spatial distribution of whole transcriptome at the single-cell resolution. Unlike the other spatial spot deconvolution method that gives you a cell type proportion at each spot, this spatial scope directly gives you the exact single cell and the cell type at each spot, because it also takes the nuclear segmentation information from the HE staining, and they locate and count the cell, the nuclear in each spot. So with this powerful tool, we now get the single cell level spatial slides, and consistently we observe this yolk sac erythro megakaryopoietic niche in multiple spatial slides. We see that it is consists of the erythroid megakaryocyte and the yolk sac even in the same spot. And the original HE staining also suggests that there might be blood generating from this niche. And similarly, the immunofluorescence staining also suggests that hemo can generate the blood island that mimic the yolk sac. We see that in the blood island, there's a lot of erythroblast inside. So we are curious. We're curious that what is happening inside this niche. So we conduct the spatial ligand receptor analysis and found that the vitronectin from the yolk sac endoderm can activate a group of integrin signalings with the vitronectin integrin alpha 2b. This pair ranks the highest. And the spatial and single cell gene expression profile also confirms this activity. And to validate our observation, we also compare the hemo data set with a public human fetal yolk sac sample. We found that the hemo can correlate well with the yolk sac sample at the cell type level, and again we observed the vitronectin expression from the yolk sac epithelial cells, and these integrin alpha 2b are expressed on the megakaryocyte and some of the progenitor cells. So this is again support our observation. A quick summary of the first part: we model the human embryonic development and hematopoiesis from the stem cells. We use the time series 10x chromium and VISIM to define the gene expression landscape within the organoid. We highlight one of the signal, the vitronectin integrin alpha 2b, this signal activity within the niche. Now we have this in vitro system that can model the human early hematopoiesis. Then the second question is, what is the developmental process inside the organoid? We first focus on the erythropoiesis population in the hemo, as it is the first indication of hematopoiesis during the development. We found that hemo covers the erythropoiesis from the pro-erythroblast to early erythroblast, and finally the more matured also chromatic erythroblast. And we see the overall reduction of CD34 expression along this trajectory. And there's also a sharp reduction of integrin alpha 4 and CD71 on the tip of the also chromatic part. And there's also increased expression of CD235A and CD233 expression on the also chromatic erythroblast. So these microgene expression profile are all consistent with the previously defined markers. However, when we incorporate the time series information, we suspect that HAMO is likely to generate more than one erythropoiesis wave. We found that in the day 8 sample, they have a higher proportion of the more matured orthochromatic erythroblast compared to the later 2 time point. The pseudotime analysis also confirmed the trajectory from pro-E to early E and finally the orthochromatic E. But when we look at the sample, it shows that the day 8 erythroid cells are at the later temporal order. This means that the pseudotime doesn't align with the real time. There might be 2 reasons. The first is that the developmental kinetics have been changed. The second reason is there may be 2 waves inside this organoid. And we also found that the day 8 erythroid cells have express higher level of hemoglobin subunit zeta, which is a marker of embryonic erythropoiesis, while the other adult hemoglobin genes are more expressed in day 15 and 18 samples. So collectively, these three evidence suggest that HAMO can generate at least 2 erythropoiesis waves. To distinguish these 2 potential waves, we further apply the Meister system to enrich the mitochondrial genome from the individual Tenix chromium and Visium cDNA library. With this design primer pairs, we can enrich the mitochondrial reads coverage to hundreds and even 1,000. With the computational pipeline for variant calling, we get a list of single nuclear variations along the mitochondrial genome that can explain the variance of the among the cell population. Surprisingly, we found that some of the mitochondrial variants can label the multiple hematopoietic waves. For example, in day 8, these 2 single nuclear variations are more likely to label the extra embryonic wave. It starts from including the erythrocyte, megakaryocyte, and monocyte. While the variants from day 15 and 18 sample are more likely the intra-embryonic wave that goes through from the hematopoietic stem cell and progenitor cells and then give rise to all the sublineages. A summary of this part, HAMO generates at least 2 erythropoiesis waves, including the both extra-embryonic and intra-embryonic lineages. And the enrichment of mitochondrial genome and confident variant calling allows us to distinguish the hematopoietic lineages. Now we know that there are 2 different hematopoietic waves inside the organoid. Our third question is, how are the cell fate decisions made in different waves? So to answer this third question, we plan to use the Larry lineage barcode to recover different hematopoietic waves. The Larry system introduced a unique DNA sequence into the cell with the lentiviral vector. And after the cell proliferation and differentiation, all the progeny cell will carry the same genetic barcode as the initial cell. with the single cell RNA sequencing, we can harvest the Larry genetic barcode and as well as the transcriptome profile. This allow us to explore the clonal relationship on this continuous cell landscape. What we plan to do is to introduce the Larry genetic barcode at the germ layer patterning stage to which has most abundant mesodome population. And after the differentiation and culture, we will harvest the time series progeny cells and do the single cell RNA sequencing and target barcode sequencing. With the computational analysis, we can then distinguish different hematopoietic waves inside the organoid and tell whether they're coming from the same mesodome and endothelial cell population and how their cell fate decision made during the development. Yeah, and finally, I'd like to thank my supervisor, Dr. Ryo Sugimura, Professor Pantaleo, and Dr. Yuanhua Huang, and all the lab members for this project. And I would also like to thank Bertie Goggins, where now I'm visiting his lab and also get a lot of help on these hematopoietic waves. Yeah. And I would like to take the questions.
Sorry, it took a
Is
little while to
Huang
unmute.
raising hand?
Yeah, I love to ask a question. It's very interesting, and it's definitely not my field. So if I misunderstand, but I'm curious about the mitochondria mutation to track the lineage. Is the mitochondria mutation linearly correlated with how much the cell divides? And so, you know, it's definitely linearly that when you have a SNP, that's a linear time in terms of how much cell is dividing, and that allows you to track that way.
Actually, for this mitochondrial sample, we should regard them as individual replicate because it's coming from individual sample, so it's hard for us to linearly align different samples at different time. But so here we are individually looking at these different samples and found that they can actually they can mark some different hematopoietic wave.
Got it. Thank you.
Very nice talk, if I may. Very nice talk, Yiming. I really liked the workflow as well as the clever integration of this master analysis to identify the waves of hematopoiesis. So that is very, very nice. Yeah, I have, as an RNA biologist, I'm really excited when people, when you compare the single cell transcriptomic data with the visualization. Yeah. So, so in this respect, I would imagine that since you're trying to distinguish significantly different diversity of cells, how many cells did you have to analyze in the VCM to compare that back to the single cell RNA profile?
I think there's no limitation of the cell number, and for these slides, the input the spot we get is around 1,200 spot, and
okay.
After the deconvolution we get like 3,700 single cells from
Okay,
this
so
slide. Yeah.
Also, there's more than enough individual data sets, data points to then to provide the separations. Okay, very nice. So, what is your idea about? What's your thoughts about this 2 waves of, or maybe even more waves of the erythropoietic waves that you found in the organoids, and how would that compare that with the embryonic development?
You mean the wave and the development?
Yeah.
So
Okay,
yeah,
let me sorry, did I make myself clear? So what I meant was, you saw these two independent waves of erythropoiesis in your organoid system, no? When you looked at the kinetics, yeah. So my understanding of embryonic erythropoiesis is very limited. That's why I was very curious in the normal development in humans, how many waves would one need to expect, and how would that compare that with the organoid system that you have, so that you can kind of place where your organoids stand in terms of the normal course of hematopoietic development in humans.
now in mouse, now it is very well known that there are two extra embryonic waves in the yolk sac and one intra-embryonic wave that is called definitive wave from the AGM. And in human, it is still not well clarified. There's a preprint from Bertie's group last summer. They collect a time series yolk sac sample and found that actually in human yolk sac, there are also two extra embryonic wave within the yolk sac. So we are now also comparing the hemo data with their human yolk sac data, see if that can be aligned together.
So
yeah.
Okay. So did I get it correctly that so you can really distinguish between the extraembryonic and the intraembryonic waves, or it is just the cycling of the erythropoiesis, meaning that you do see multiple rounds of erythrocytes being made within the same wave, or these are individual separated events?
Oh, that's a good question. Yeah, that's a very good question. So I learned from the paper that after so when the erythroblast goes into the terminal differentiation stage, and it cannot they cannot proliferate again. So it is a single way to
No, that's not what I meant. So we have proerythrocytes, no? So but is it always very well synchronized that you always find the same? If you catch the cells in one place, it will be always in that wave, or there are multiple proerythroblasts, and then each of those populations will cycle in its own cycle. You know what I mean? I'm not asking whether the erythrocytes can give rise to proerythrocytes, but you see different waves, no? So is there individual differentiation cycles of the proerythrocytes, or these are individual waves of hematopoiesis? Okay, forget about it. we can talk about it afterwards.
Yeah, we can talk later.
Sorry, I think I confused you with my question.
Yeah, but I do observe that this wave, for example, this wave, it appears in the tip of the UMAP, and it is the also chromatic erythroblast, so it goes quite very quick, and
okay.
For this part it is slower. Yeah, so it's likely this is the first wave, and this is the second extra embryonic wave.
Very nice. Maybe we can go on to somebody else for questions.
Yeah,
if anybody has.
So thank you very much, Jimmy. This is a great presentation, and I really also naive on this topic. I do like hematopoiesis, but I'm really like naive. So first of all, for how long could you do like the organoids? For how long could you stay the organoids in the lab?
In my memory, it's up to at least 5 or 6 weeks.
Okay, and so. Yeah. I mean, I work with mice, so I guess in humans, it's very different, but regarding the timing, but could you like, in this in this protocol, could you like find some of the precursors of the hematopoietic cells that will go to the liver? Like, could you detect them? Like, if these precursors will eventually in the human will go to the liver, or if you could map those precursors that will give rise to T cells or lymphocytes? Maybe.
Yeah, that's a good question.
I know that of course you will find T cells there now, but maybe the precursor of you could do some ATAC-seq to look up some genes that will mark the T cell lineage in this model.
Yeah, that's a very good question. So we also have another day 32 sample with a lymphoid culturing system, and we can get a group of lymphoid progenitor and also some of the mast cell and NK cells. So Rio will cover this part of the content in his talk, and yeah, they are functional cells.
Okay. And just like really a naive question, in the cell culture, how many organoids do you have? I mean, you have a single cell in the plate, only one organoid, or it's more than one? I'm thinking about maybe the signaling that could have between the organoids in the plate.
For each sample, we collect like 5 to 10 organoids and mix them together as a sequencing sample. Yeah, but indeed there might be some also some interaction among them. Yeah, some cytokine.
Yeah, I was thinking about that. Thank you.
Thank you.
Hi, Amy, great talk. Can I ask a question about speed, time point of this organoid? So I'm surprised you can observe the hematopoiesis at day 8. It's so quick because and so I'm not sure how long you can see this kind of event in vivo in human embryos.
If you're sure. For the hematopoiesis part, at least to day 32, we still have this hematopoietic population in the sample. And if you're curious about the erythropoiesis, they may stop at around day 25, like that.
So do you
we
sorry, go ahead, please.
Yeah, and I think this hematopoiesis happens quite early when we culturing this organoid. There are releasing cells around the organoid, and when we do the FACS, we found that they are actually the blood cells releasing from the organoid. So it is very likely similar to the yolk sac that they pumping the cells out. Yeah.
So do you add some specific cytokines or inhibitor or something to induce differentiation in organoid formation?
So we use the stem-diff conventional medium to ensure that the reproducibility, and we didn't add additional cytokines except for the original kit. Yeah.
So that means this kind of differentiation was induced by the intrinsic stimulation or pathway. So what kind of mechanism? So do you think this kind of differentiation was induced by the just cell-cell interaction machinery?
Sorry. That's I think there's still cytokine in the stem-diff commercial medium. Yeah, but we just don't add additional one on that.
Okay, and the last question is Did you try with conventional human ES cells to induce this kind of organoid?
Not really, but we compare our single cell data with another paper. They use the ESC for the differentiation, and they also get the extra-embryonic wave and intra-embryonic wave, and actually they integrate quite well.
So that means this kind of expanded potential stem cell is not definitely necessary.
I mean, there's still difference. Yeah, there's still difference.
In their efficiency, you mean?
In the cell population, because it's not totally overlapping.
Okay, thank you very much.
Yeah, thank you for the question.
If there is any not any more questions, thank you very much, Yiming. Very nice talking to you. It was very clear, and really excited to see what's coming out of the next part of your adventure. If you can unshare the screen. Now, next, I would like to introduce Rio. Rio is currently an assistant professor and a PI at the Center for Translational Stem Cell Biology in Hong Kong and at the University of Hong Kong. He did his PhD at the Stowers Institute, and before that, MD in Osaka Medical University. Among the accolades, he's a receiver of many prizes, including the Genius Prize at the Young Hematologist meeting in Japan and March of Time Scholarship. In his career, he has contributed consistently to the field of hematopoiesis, and he's one of the early adopters of combining wet lab with dry lab, and for taking our understanding of hematopoietic development at the systems level forward. If I may point out two very key contributions Rio has done to the field of hematopoiesis. He unraveled the non-canonical Wnt signaling and its importance, and it's important in the context of human hematopoiesis. And in the lab of George Daly, he developed methods to generate hematopoietic stem cells and progenitor cells from human pluripotent stem cells. And Rio, take it from here.
Yeah, thank you very much. Yeah, wonderful introduction. And yeah, amazing talk by Yiming. Yeah, so this is real. I am PI of HKU. I'm nice to see my best, all friends. I got a bit pneumonia, so probably my voice can survive 10 minutes. But I'm sure Yiming has already done an amazing job introducing her new work. And I'm going to do is basically catching up what Yiming showed. Basically based on her chemo, those organoid, what we can do, and what is another focus of our lab, because Amy and her team has been addressing fundamental questions. As already you see, like a lot of lineage barcoding, addressing question of cell origin, because some people always, take their origin. And then, what we can do from, beyond that, what we can do from more practical thing. So yolk cell. This is amazing organ, and they can produce NK cells that I briefly touch on this talk, and tissue resident macrophage, everyone respect, via microglia, because this is very important for neurodegenerative disease. Some type of minor T cells, type B1B cells, of course, RBC, and blood vessel. So this is our chemo, if you see by microscopy, and also my background of this. So I'm using this one, trying to see vascular network. So that's exactly one of my MD students here is modeling COVID-19 vasculitis by infecting the poor organoid with real virus. We try to understand how you can enhance tumor organoid by fusing this guy. So essentially, this actually majority of our team in the lab called this is vascular immune organoid, vascular immune. So that's basically donor of blood vessel and immune cells like macrophage, NK cells. If you fuse with any of tumor organoid, So that's why, our motivation comes from trying to, maximally use this chemo. And of course, this guy can produce NK cells and macrophages. I will be actually this talk touching on some NK cell part that published by our undergrad, and macrophages is our major product. All right. So yeah, thanks to our undergrad, Angie Poon, she already graduated and went to industry. Amazing career. So yeah, actually that's why today I am here to cover her paper. But she has done amazing job, including setting up system to generate functional NK cells from means hemo, and basically very much. Yeah, still working on efficiency, but kind of be satisfied. We can see like activation marker, like degranulation, CD107A, tumor killing capacity. So this is already published. I'm sure protocol, video protocol is available. I am just working experiment. So thanks so much, Angie, and you have done amazing job. So this is also, why NK cells? Why NK is important? Because that's a source of new type of CAR immunocells. CAR T, everyone knows. CAR LK. It's quite, highly appreciated new type of CAR immunocytes, like Katie Rezvani using cold blood and producing CAR NK. Already been to clinical trial. It's amazing result. They have high cytotoxicity against cancer, but less GVHD being safe, probably way safer than conventional CAR T. And Dan Kaufman leading this field using iPS cells, generating CAR NK. And NK is random killer, but we can use CAR to endorse specificity. So asking this CAR NK to target specific types, namely cancer. So what is current stage of CAR NK? This is already like more than 5-6 years concept, and many amazing papers and actually saving patients. Basically, a CAR structure that is binding tumor antigen and artificial chimeric receptor and provoking NK cell signal. So that's basically essentially exactly identical to what we see in CAR. Other people are more like playing a lot on synthetic biology, like having cytokine expression, armored CAR NK or aggregate for more safety. And obviously, important feature of this CAR NK is you can take advantage of cytokine biology because we have a lot of knowledge of cytokines that specifically activates NK cells. And you can force express or you can specifically induce in CAR NK. So basically ensure the high capacity expansion and basically safety. So although Angie already been to her own amazing career, but actually my love keeps NK cell part. Sanshin, another PhD candidate, second year, he has been continuing this part, and every day he is busy making NK cells from human PSCs, introducing CAR's construct. Basically, I'm going to touch on more on Sanshin's recent work, ongoing work, like how to use immunopriming cytokines and ideally in membrane-bound cytokines that enhance survival and expansion of CAR NK. So basically, CAR NK has a clear direction how we have to move on. So this is our, I would say, core team. Basically fundamental science, she's amazing. She already, making her own group like think, basically gathering mathematician and collaborators, informatician, and very independently already working on her own projects. And I will be looking for more papers from her, fundamental biology. Amazing. So this is my practice. Joined by Patrick from Italy and Sophronia, local student, Theo from Heidelberg. And Sanchin, that keeps mentioning, is amazing PhD candidate. So now, chimeric antigen receptor macrophages. Why macrophages? So let me start why my lab is so much on to macrophages. So I mean, everyone knows CAR-T is important, like CD19, B lymphoma, targeting CAR-T cells. But one of the major challenges of CAR-T is solid tumor. This one cannot go into like inside of tumor. Maybe I have to put some like laser pointer here or basically like offset poor. I'm terrible for CAR-T. Interesting. What's inside of this tumor? Majority is macrophages. So, basically, it's basically brief idea, very primitive idea. Can we take advantage of those macrophages? Very happy to be or migrate into tumor, and we can exploit them to be cancer enemy. So that's how idea of CAR macrophage came from, and Sao Zero in UPenn, and basically they published amazing paper using primary macrophages introducing CAR construct, and basically show specific cytotoxicity, especially enhancement of phagocytosis against cancer. And of course, in vivo works amazing. So this is a CAR macrophage. I mean, our motivation, and the field is very busy and expanding its universe. CAR T, NK, macrophage, probably DC, gamma delta T cells, many other type of immune cells are joining. But essentially, what we have to understand is each immunocyte has their own pros and cons. Like CAR T is still amazing for lymphoid tumor or leukemia. CAR NK for its safety, like less ZVHE, this is highly appreciated and already shown in clinical trials that we can learn a lot from patients there. CAR macrophage, their own venue, I mean, unique site is its migration capacity in tumor. So that's why, I mean, my lab focus on macrophages, especially CAR macrophage to endorse cytotoxic capacity against hepatocellular carcinoma, liver cancer, because in Hong Kong, this is our social demand. We have to address this challenging cancer invading in East Asia. So macrophage, actually, this is the oldest immunocells. If we go into textbook or immunology and like review, actually, this is one of the oldest cell type and comes first in vivo, I mean, embryo, already mentioned by Amy probably, and yolk sac, we can already see macrophages, amazing. Technically, this is our best friend because if you use iPS cells, I mean, any type of pluripotent stem cells, you can see macrophages after 2 to 4 weeks culture. Then probably neutrophil, NK cells, and don't ask me T cell. That one takes 1 to 10 weeks and still not much consistent in the available protocol. So yeah, that's why I would say macrophage is our, I mean, best partner to with cancer, especially people playing with iPS cells. All right, so yeah, this is our motto. I mean, entire half branch of my lab in science part, I mean, my second lab, focused on cancer immunotherapy. We have to understand 4 key criteria for cell therapy. One is locomotion. Can these immunocells go inside the tumor? And that's macrophage, nice to have. Secretion. If we want to secrete payload like cytokines, single chain FV, your immunocells have to be good at secreting protein. And we know T cell is amazing because their cytotoxic capacity by interferon gamma, TNF production. NK good because they also making powerful granzyme. Macrophage, I mean, we are working on. Probably we have to engineer a bit. And resilience, like sustainability and viability in vitro and in vivo. I mean, T and K already people show that in patients. And macrophage, yes, I'm going to show some new data. Process, I mean, since I am a human iPS person, over 10 years working on this system, I always care like reproducibility, time, cost, safety of the procedure. And macrophage, as I said, that's the easiest one, amazing one. Don't ask me this. So this is basically macrophage, monocyte to macrophage differentiation, like very easy scheme. Like you put certain cytokine combination, and yeah, the stem deep is amazing. That commercial kit has a bunch of, or maybe tons of cytokines. And we have like a rough idea what kind of cytokines they have. Basically, those like GM-CSF, MCSF, and then we can add on interferon gamma, LPS for more inflammatory macrophage. My lab basically more focus on this left side, M1 macrophage. So always, every day we are skewing macrophage into more inflammatory phase. So that's what we do. So again, this is hemo, but more like a modified protocol of this hemo, and you can input monocyte medium, macrophage medium, and then from sensory technologies and bunch of cytokines in the kit. Very expensive, but robust. And you can get macrophages, like you can see in this picture, like by cytospin. Amazing job by Sanshin. And also you can stimulate it by inflammatory cytokines, and you can see still into pro-inflammatory effect. Or vice versa, like anti-inflammatory cytokine can still into anti-inflammatory effect. All right. So, what is a function? Then, I keep saying like macrophage easy to make, but of course, people always ask us, the functional, like, same as true bona fide macrophages. And yes, yeah, they can do E. coli, phagocytosis, and they're actually expressing HLA-DR, antigen presenting molecule MHC class 2 very robustly. And they express phenotypic markers of macrophages like 45, 11B, and more other too. They are coming to one of the point I introduced resilience, like basically macrophages. One of the challenge of primary macrophage is its resilience. You could culture primary macrophage over weeks in much some cannot expand, because they are already mature and wouldn't cell cycling. So I asked Sanshin, hey, can you keep your culture for 4 weeks and see what happens? Very rough idea. But yes, actually after 4 weeks, even still we can see expression of M1 inflammatory markers and M2 anti-inflammatory markers. So they basically kept its phenotype over culture, as well as high viability. So that's basically encouraging this, like using those stem cell progenitors to macrophages, you can avoid typical issue of primary macrophage. And of course, gene delivery is one of the major caveat of regular primary macrophage. You have to use a very particular type of adeno-associated virus for primary macrophage, limited access to many labs. But with using IPS cells, EPSCs, CRISPR-Cas9, very easy, and you can also do like lentivirus and PDBAC. So let's go into what we do, like trying to enhance, trying to go beyond the regular color macrophage. So there are two approaches engage macrophage killing cancer cells. Like one is taking advantage of immunopriming cytokines. So like this macrophage can basically enhancing the immune cells around those macrophage. Immunocheckpoint, especially like monocyte immunocheckpoint. CD47 is a very famous target. So essentially, if you target CD47, you make your macrophage big eater. They will eat a lot of your cancer. So basically two approaches, major method to enhance your macrophage. So yes, Sanshin already took this approaches, and he verified immunopriming cytokine like IL-12, 18 can enhance cytotoxic capacity of NK cells, as you can see by enhanced expression of interferon gamma in vitro. So that's basically telling, hey, if you put IL-12 or IL-18, probably expressing from your current macrophage, these macrophages will be able to immunoprime surrounding NK cells. Now remember, we can make NK, we can make macrophage. So one shot, you can activate both. Then CD47 sub, this is done by amazing job by Rami Majetti and Av Weissman originally, like nearly 10 years ago, and a lot of attention into this pathway. It's one of the major checkpoint in monocyte macrophage. I mean, a bit complicated, so let me walk through. I mean, basically, cancer cells express CD47. Your macrophage express SIRPA, and they bind, send a signal, and silence your macrophage. So this is how cancer cell, escape from macrophage. Very smart guys. So idea is if you silence SIRPA or macrophage, and it's in vain, cancer cells cannot silence macrophage any longer. So basically easy genetics work. Not easy. He has been working on this gene targeting for a while, but recently got an interesting result. So already, Sanxin verified his macrophages from EPSCs express SARPA high level. You can target, genetically knock out the SARPA, putting carb, probably like a P2A conjugating or cleavage and IL-12 or IL-18. So you have basically two ways. So two-in-one approach. By this strategy, you can basically take advantage of immunopriming cytokines and SARPA inactivation. So basically, I show my one of the major branch of laboratory. Yeah, and basically we are exploiting cytokine biology. Example, we convey immunopriming cytokines, and we want to broaden cell potential because that's the advantage of using iPSC, EPSC, easy genetics. So you can engineer any loci you want, and we pick up SARPA as one of the example. We have another story in PDL1, but that's more mechanistic one. So yeah, I would say SARPA is more relevant to today's talk. So my lab there, we have two programs. Our blood develops, and the other team, synthetic immunology, especially trying to broaden counter immune cells. And right thanks to Angie, our undergrad student, set up NK cell culture, published a paper with us, and taken by Sanxin. And basically, Sanxin is teaming with Theo, Sophronia, and Patrick. So that's all I have today. Thanks so much.
Thank you very much, Rio, for the nice talk. It's time for questions, and maybe if nobody else is asking, maybe I can start off. You showed this really nice transition and of the appearance of M0, M1, and M2 macrophage populations upon a longer-term culture. Do you know if these are independent populations emerging in different time windows, or are the M0s transitioning linearly to M1 and then M2?
So you mean this part, right? Like M0, M1, M2 thing, yeah.
But when you also had your culture conditions where 4 weeks you saw these populations fluctuating. So
so we still anyway, if I want to have M1, I just add interferon gamma and LPS in culture as well as GM-CSF. That lose M2 capacity. So it's kind of a choice. You have to go either.
So it is not the M0 that becomes M1 and then M2. It is different populations of cells.
Exactly, two branches, right? So that's why, this is also interesting point of macrophage biology. Initially, people try to exploit M1 macrophage, and even in patients, injecting into tumor. But this M0 to M1 is a bit unstable unless you genetically engineer. Even if you go into some immunosuppressive environment, easily lose M1 capacity somehow, and they go into some exhaust, weird type of macrophage. So that's also primary reason why cow macrophage using CD3 zeta, that's mimicking toll-like receptor 4, essentially keep activating M1 direction.
So in your case, when you had this 4-week culture, you saw the 3 independent populations emerging, no? In the later slides. So would that mean that each of these populations were existing, but they amplified in different windows of time in your organoid system? Oh, okay. Very nice.
Yeah.
do we have any more questions?
Hi, Rio. So I would like just congratulations of your work, and I would like to ask you how stable are these macrophages, because there's some data that they're like you cannot divide M1 and M2. It's like a cloud of transcription between that.
I know, yeah.
I don't know. I mean, all of those
Yeah.
works are done with
My favorite. So actually in my lab, I ask every single student, don't call macrophage M1 or M2. Because that doesn't make any sense. If you do single cell, you see 5 to 6 populations. So I mean, I just use M1, M2 thing as a jargon,
Okay.
like an easy going jargon. But for real paper, I have to come up like better, distinction.
Okay. And how, I mean, I'm thinking of these used in the clinic. How stable are these macrophages, like the ones that produce interferon-gamma, if you put them in a, I don't know, TJ beta condition or something that it will immunosuppress, even though they have the CAR T, the CAR molecule.
That's, very fundamental question. I'm not addressed in CAR macrophage field yet. Obviously, $1 million question is you can put CAR macrophage in tumor, and essentially this is immunosuppressive. So where and what happened to this CAR macrophage? No one, in my knowledge, addressed that. Nowadays it should be easy. You take as Emi did, you can take spatial transcriptomics. Obviously, single cell thing. Chromium is big challenging in certain population microphage because they are eating each other. But still, 10x vision or higher resolution, like stereo-seq, you'll be able to address what happened to this macrophage. Because CAR T field, we already know this CAR T has been abused by cytotoxic cells in the tumor. So in this same situation we are having, or CAR macrophage or steward, no one knows.
Thank you.
Yeah, I see my old friend. Yeah, she paid. Long time no see, like probably 5, 6 years ago. Yeah. Can you unmute?
Yep. Okay. Yeah. Hi, Leo. A great talk. A long time no see. I'm really happy to see a great talk today. So I'm just curious, how much efficiently you can induce these macrophage or NK populations, and how similar they are to compared to in vivo cell types, at least in
Yeah, thanks for my
transcriptome.
favorite question. So for macrophage, I have easy answer. So I let other people, especially clinical department in our lab, try our protocol. And they are already producing macrophage using our protocol. Comparing like a very bona fide real macrophage for expression and function. Very similar. They say, oh, couldn't distinguish so much. And okay, good point. I have to say, probably, if you go into more sophisticated way in this side, on right side, you become more tricky. T and B always very challenging. NK, this is really iPSC, NK, more like innate lymphoid cell, ILC. So some people call like a ILC NK such a jab, but because you couldn't define like these things, the bona fide NK, so that's why they come up some weird way of coding this poor population. But sure, you have to engineer this NK, Dan Kaufman does successfully introducing CAR. I believe putting those toll-like receptor mimicking CD3 zeta, you further enhance cell fate. Like NK will be more like NK if you do this engineer.
I see. Okay. So how about the cell number? If you want to apply to the clinical trial, you need to expand
Nice.
the scale.
Nice. I think this is also my favorite question, when everyone asks me. So basically I have to say, if I ask a postdoc produce, hey, can you try big batch? And she will come up hey, I got a 6 million macrophages. And of course, this is only one batch. So if you combine multiple batches, more plates, human source, you will be able to get more number of macrophages. NK challenging, T more difficult. Actually, my lab also does T and also NK, but Yeah, always I have to look for some better way to expand NK cells. But actually, this is already done by Dan Kaufman's team. So yeah, I am more optimistic team. I am not good. Macrophage, yeah, easy.
Oh, macrophage sounds promising. Thank you very much.
Yeah, thanks. It's my voice survived for about 10 minutes. I thought I wouldn't be able to speak much today, but yeah, nice. Okay. So probably eventually we call it a day, right?
So thank you very much, Rio and Leo and Yiming. So I would like to take the time to invite you to the next seminar. We're gonna talk with Maria Cecilia Campos from the University of Rockefeller, who she's using a really interesting mouse model, where she basically can detect all their interactions between the cell live. So it's a reporter mice. It's really interesting. So if you could join, I think this technology could also be applied for hematopoiesis as well. So thank you very much for joining today, and congratulations all of you. Bye bye.
Yeah, bye. Yeah,